Good morning and good afternoon, everyone. Welcome to our webinar, Introduction to Single Cell Multi-omics Data Analysis and Live Demo. I'm MJ Kim, Marketing Manager at Mission Bio, and I'll be your moderator today. Our speakers today are Kelly Kaihara, who is an Applications Manager here at Mission Bio, and Heatmap, who is a Field Application Scientist also at Mission Bio. Before we get started, I'd like to let everybody know about this upcoming webinar next week, hosted by Covance, where we'll be joined by industry experts on oncology drug development to talk about single-cell multi-omics. You can visit our website at www.missionbio.com to learn more. Next slide, please. So a couple of reminders. We're currently recording the session and streaming live on YouTube. Also, at any time during the webinar, you can type in your question in the chat box, and we'll ask the speakers during the Q&A session after their presentation. Okay, and with that, I'd like to turn it over to you, Kelly. Please go ahead.
Hi, thank you, MJ, and thank you for everyone for attending. So we're really excited about launching our new multi-omics capability with a commercially available kit in conjunction with BioLegend. And so the purpose of today's seminar is really to show you how to unlock the data and use our software and data analysis tools in order to analyze these very complex multi-omics data sets. So we'll do an introduction to data analysis, including a live demo with our Tapestri Insights software. And it will be me and Hema for this presentation. Next slide. So multi-omics is hot, especially on a single cell level. Nature Methods called it the method of the year in 2019. And I think it's really exciting. Single-cell RNA-seq, the first commercially available product in a high-throughput scale, came out in around 2016. And we're starting to layer more analytes, including single-cell DNA-seq, ATAC-seq, protein-seq. And so it's just been like a really interesting and beautiful phenomenon to follow how this field has exploded in the last 5 years. And we at Mission Bio are really excited to be a part of this explosion of new technology. Next slide. So why do we want to shift towards multi-omics? Well, we want to get as much data as we can from very precious patient samples. So like I mentioned in the previous slide, we have many new cutting edge technologies such as the epigenome, genetics, the microbiome, the proteome, the transcriptome. And so with the advent of all these new technologies, typically we want to run our sample through as many of these as possible, but then we're left with the task of very complicated data integration. So how do we stitch together all these pieces of information from all these different analytes back into a meaningful signature so that we may better provide precision medicine to patients with disease? And so this is hard when you're doing, when you're running that sample across many different platforms. Next slide. And this is illustrated in this schematic. So you might take that sample from the patient and do bulk NGS to look at single nucleotide variants. You could do flow cytometry to look at your protein markers, maybe run a CMV array, and then get single cell RNA-seq data. And again, you're splitting that sample across many different technologies and then needing to stitch back all that data together for each sample. And so really, what would happen if you could get all of this information from a single sample at a single time point and get more out of that precious sample? And that's what we aim to do at Mission Bio with the Tapestri platform. So we're layering on more and more analytes. We started off with DNA and SMVs. We recently included copy number variation, and now we're moving on to protein expression. And so from that same single cell, you now get information across many different analytes and from thousands up to 10,000 single cells per sample to boot. So the amount of information that we're getting from a single sample has astronomically increased. And so to tackle all of that data, we've now switched to H5 files, which can handle information across all of these different parameters. And we've worked really hard in the past couple of years to create software tools and data analyses packages in which to analyze these data to make it easier for the customer to very quickly get very beautiful illustrations and visualizations of what's happening across all of these different readouts. Next slide. So Mission Bio offers a complete end-to-end solution. We have all of the assays in order to look at DNA plus protein. We have the instrument and the microfluidic chips in order to accomplish the ability to put cells and droplets in a high throughput fashion and get to the DNA and the protein signature for thousands of individual cells. And again, we worked really hard, and this is what most of the presentation is going to be about, which is data analysis, the really exciting graphical depiction of what is lying in your sample. Next slide. Here is an overview of all the offerings. So we provide if you would like to do DNA plus protein, we introduced a new stealth staining reagent kit shown in the black and blue box. And that is a simple 30-minute protocol that goes upstream of your current single cell DNA sequencing that you would do on the Tapestri platform, which is illustrated by the DNA core reagent kit. So again, just a simple add-on if you want to shift from doing DNA only to DNA plus protein. And then again, we have a new cloud computing access and analysis solution, which is able to now do DNA plus protein together. If you're interested in DNA, so SNVs and CNVs, we have a number of catalog panels across hematology and solid tumor space, or we allow the ability with our Tapestri designer software to customize those DNA panels and add targets of interest or subtract targets that you don't need. In addition, in conjunction with BioLegend, we offer a 45-plex for the hematology space, as well as custom antibodies that you can get through Mission Bio and BioLegend. And then on the right here is our software solution. Tapestri Pipeline does the secondary analysis. So primary analysis is done on your Illumina sequencer. Secondary analysis, you need to take those FASTQ files and line them up to the genome and deconvolute all the cellular barcodes. So that's done with Tapestri Pipeline, both for DNA and protein. And then we offer Tapestri Insights software to do tertiary analysis, where now we get to take all of those reads across all of those cells and interpret them into biological significance. We offer Tapestri Insights, which is a desktop GUI software, as well as Mosaic package, which is Python code in order to do more advanced analysis. And so today we'll really talk about these two software packages in depth, Tapestri Insights and the Mosaic package with a live demo. Next slide. All right, and with that, I'll hand it over to Hema.
Awesome, thank you, Kelly. Thanks, everyone. So yeah, as Kelly said, we are extremely lucky and very appreciated of the work we have done together with Dr. Ross Levine for the Multi-omics application note, which was the first application note we launched for this Multi-omics assay. So some data of this application note is what I will show today for the live demo of Tapestri Insights. So just to give you a little bit of background, this application note has two different experiments. One of them is to showcase the 45-plex total seq-D cocktail from BioLegend plus PBMCs from patient samples. And the other one is the one I'll show you on the live demo. So what I want to show you first is a couple of images from this application note, just as a sneak peek of what the software Tapestri Insight and Mosaic can do. So the first one is just, just a screenshot of Tapestri Insights 3.0, where you can see a mixture of PBMCs from 2 healthy donors that have been analyzed with a total-seq hem-oncology cocktail, which is 45 plex, so 45 different antibodies, and a myeloid panel from Mission Bio, so the myeloid DNA panel from Mission Bio that contains over 300 amplicons and targets more than 50 genes. And again, I'm not going to focus on these pictures because I will show you live what Tapestri Insights can do. Then the second part of the app note highlights Mosaic. As Kelly said, this is perfect for advanced bioinformatic analysis. So here what you can see is a heat map showing the greater resolution of the PBMC subpopulations. As I said, there are 2 donors, PBMCs from 2 different patients, and 45 antibodies at the same time. So you see a great resolution, and you see very nicely explained the results of the 2 different individuals thanks to the resolution of the 45 antibodies. So that's one part of the app note, which we can share with you after this presentation. It is also available on the website. But the other part of the app note is a small data set, which is 1 sample performed with custom DNA, a panel, and 6 antibodies. So now I will stop this presentation, and that's what I will show on a live demo of Tapestri Insights. So as Kelly said, Tapestri Insight is a very easy to use GUI software package that you can just download and install on your laptop. So I have here a window just to show you how the home page looks like. So you have the option to start a new analysis, and the input has now switched to H5 files. So you can see here your different samples, and at a glance, you get the total cell count and the panel that was used. And you have already, you have links to the website to see a quick walkthrough if you need, a quick video to help you do the analysis, the release notes for the software, advanced documentation, and the support page. So after you upload your files and start a new analysis for the H5 files, this is the window that you obtain. I just haven't opened to make it easier. So that's the first thing you see. You have your sample 1 with the 2 analytes, DNA and protein, number of cells, and then you have the advanced filtering option. So for those who have used the previous version of Tapestri Insights 2.2, they might be familiar with these advanced filters. So I'm not going to go into much details for these filters because they remain unchanged from the previous version. But just so you know, this allows you to change different parameters in order to filter out variants or cells. As an example, you can change the genotype quality or the read depth. We have additional information on these advanced filters in a video that we have posted on our YouTube channel, and we also have additional knowledge base articles for those who want to know more what's happened behind the scenes for these filters. For this demo, let's just now focus on these 2 windows first. The first one is the review variants. And here we can see the list of the variant candidates. So we have a total of 27 variant candidates. For these variants, we provide a lot of information, as you can see. So you have the variant name, the function, the protein for those who have a protein, coding impact, you have information from ClinVar, you have information from DAN. DAN is a pathogenicity score. So the closer the number to 1, the more pathogenic this variant is likely to be. You also have the percentage of genotyped cells, and from these, the percentage of mutated cells. Then by sample, you also have the information of allele frequency by cell count and by read count. For the review protein window, so far you can see the 6 different antibodies that were used to analyze this sample. So now, in order to continue with our analysis for this specific demo, we will focus only on the pathogenic variants, which we know now, and we can sort descending by ClinVar that are IDH2 and DNT3A. These 2 pathogenic variants were also confirmed by bulk, and these are the ones we are using today. So after we have looked at our data, see the variants and the proteins that were used, now I move from the filter window to the explore window. So as you will see, the software is doing something in the background. So what the software is doing is clustering the data using dimensionality reduction techniques and projecting that into UMAP space. And that's what you see here in the main window. But before going through the main window, let me show you these different menu bars, which are completely new for this version. So as this is a multi-omic assay, you can choose, of course, the source for clustering. You can cluster based on DNA, and for that you can choose either allele frequencies or genotypes. And of course, you can cluster based on proteins. So for proteins, we have two options: Assay normalized counts and also CLR normalized counts. You see that the allele frequency for DNA is the default. Then you can also color by subclones, but by many other parameters. So you can color your clusters based on samples, DNA parameters such as genotype, read depth, genotype quality, allele frequency, or you can also color based on proteins. We will see this as we go along with the demo. For this specific clustering analyte, so based on allele frequency, we have additional clustering options. In this case, it's dbScan, and we have these two parameters as a default. So this is for you to change if you want to modify the number of clusters. You have epsilon and similarity. In this case, we will not change these parameters for this demo, and we will continue with the custom with the recommended values. Now you can see below, in case you are working with multiple samples, you can deselect the samples. In this case, we have only one. And below you can see the subclone tables. Now here on this part of the window, we have the variants. So clearly you can see the only two pathogenic variants that we have selected. You can also see the 6 proteins that we have and the different subclones. And here on top, you have different visualization methods. So we will go through them as we go along with the demo. We start by the UMAP. So now let's just focus on the main window. So as you can see, our data have clustered into 8 different clones. And you can see that some clones, like this one, appear to be artifactual. And this is completely expected, or it is normal when working with low density space, because we are only working with 2 variants here. So, of course, the phenotype and the number of clusters will change as you increase the number of variants when working with more complex data sets. But this phenotype is expected when only selecting 2 different variants. So now you see 8 different clones. They are ranked always based on absolute number of cells. So C1 is the largest clone with the maximum number of cells, and C2 the second most, and so on. So the next logical question here is to identify what are these clones. And we will start by looking at the DNA information. So for that, I'll just color by genotype. And here I have the option to color simultaneously the genotype for the DNT3A variant and the IDH2 variant. So here you have the legend for the genotype. You have the wild type, heterozygous, homozygous, and missing data. So remember I said there were some clones that seem to be artifactual. So actually these ones happen to contain missing data, as you can see here. And this legend is clickable. So what you can do to not be distracted by missing data or keep only the clones with the complete data, you can select missing and you will not see the clones that contain this missing data. So based on genotype, what you can see here is that this clone, the large clone 1, seem to be double mutant, right, for DNT3A and IDH2. And same, this clone, purple one, seems to be wild type for these 2 variants. So you also see additional clones like this one here that seems to be a single DNT3A clone, and also this one that seems to be a single IDH2 clones. So we have additional visualization methods to analyze further whether these clones are real and potentially founder clones, right? Because it is expected that you can see a single clone, a single mutant population that acquires an additional mutation. So in order to explore further these single clones, we have a Violin plot up here. So for those who have used Tapestri Insight 2.2, you might be familiar with this Violin plot. Let me just show you quickly something that you can do. You can color your subclones and look at parameters such as genotype quality, read depth, or allele frequency. So this Violin plot will allow you to study further these individual clones and see whether they are real or they are artifactual clones. For the sake of this demo, I'm not going to give you additional information on this window, just so you know this is available. And perhaps we can have a different session or a follow-up session to go more in depth for these filters. So back to the UMAP space now, we know that we could potentially analyze these single clones further, but in this case, I will only focus on the 2 larger clones for this quick demo. So what I will do now is I will name subclone 1 and subclone 2 here on the table. So subclone 1 will be my double mutant, DNMT3A, IDH2, and clone 2 will be my wild type. So as I said, for this demo, I will deselect the rest and I will focus on the double clone and the single clone. So something else that you can do on the DNA part is representing your clones in a bar plot. So you can show the percentage of your samples that each of the clones represent. And if you had multiple samples, of course, you could also do a fish plot. In this case, as we are only working with one sample, you can only do a bar plot. So that's in terms of DNA. Coming back to the UMAP now, the next question that we have is, as we have the additional analyte at our disposal, what are the protein expression profiles for these 2 genotypically distinct populations? So this is the next thing that we will do. And in order to do that, we will just color our 2 clones with the protein CLR normalized counts expression values. So here simultaneously, you see the 6 antibodies that were used for this assay and their expression and comparison between the wild type and the double mutant clone. So just as an example, you can see that the expression of CD3 seems to be higher in the wild type population, much higher than the double mutant population, and CD38 seems to be higher in the double mutant population than the wild type population. So this is the first time we see DNA and protein information together. And remember, these populations have been defined based on genotype, clustered based on allele frequencies. So the next thing that we will do now is use a different clustering method. So we will move now to cluster based on protein. In order to do that, I will use a protein CLR normalized counts. So again, the way we cluster is identical. We are taking the information, clustering using dimensionality reduction techniques, and projecting that information into UMAP space. Now, you can see here for the protein CLR clustering method, you have an option to change the number of principal components. So 10 is the default number for principal components, but we know that 10 is more suitable for larger data sets, perhaps working with 20 plus antibodies. But remember, for this specific sample, we only have 6 antibodies, and that number is quite low. So we have determined that the optimal number of principal components for this data set is 4. You might have noticed as well that the options for the clustering options, when you do a cluster based on protein CLR, now change to k-means or log-n. You can choose between k-means and log-n. When using k-means, which is the one we will use, you also have a default number of clusters of 5. Now, this software is an exploratory tool, and the analysis is hypothesis driven. So this means you can come back and forth and change the clusters based on the data that you get, the data based on biology that you get from your data, from your samples. So in this case, my hypothesis, at least at this moment, is that I have at least 2 clusters based on genotype. So what I will do is I will modify the k-means clusters to 2 to do a preliminary analysis, and then I will see whether I should increase the number of clusters. So as we can see here, when we do k-means and change the number of clusters to 2, we see 2 clones now. We see clone 2, sorry, which is the orange one, and a large clone 1, which is the blue one. So now the next step is to overlay the genotype information and see whether there is any correlation of the genotype and these clusters. Again, these are clusters based on protein data. So I will now overlay the genotype information, and we can see clearly that majority of the wild type cells do cluster with subclone 2. and majority of the mutant cells cluster in subclone 1. So again, you can see that for subclone 1, there seemed to be additional heterogeneity, right? We can see that it is not a nice, huge block. There seem to be 3 subpopulations, right? And to be honest, this is completely expected because now we are clustering based on protein, and we know from biology that the clusters based on protein do not have to correlate with the clusters that we identified with DNA. So that's what I said. Like, I started with an hypothesis of 2 clusters, but we will see whether, you know, this number should change in terms of based on the information that we acquire down the line. So far what I can see is that subclone 1 seems to be a little bit more heterogeneous. So the next step now is to overlay the protein expression of my 6 different antibodies and see how well that correlate with my clusters. So again, we see a simultaneous representation of the protein expression of the 6 antibodies. And you can see again, a high expression of CD3 correlated with my wild type cells. But look at this one, for example. So here, there seemed to be potential subpopulation, right, of clone 1 that is CD11b negative. At the same time as we see a slightly higher expression of CD38 for this specific part of clone 1. So what this is telling us is based on the information of the evidence that we are getting after assessing the protein expression, we can perhaps go back and change the cluster number from k-means, you know, from 2 to 3 or to 4, and continue analyzing the data. Again, I mean, this is hypothesis driven, and you can, you know, change it based on the results that you're getting. For the sake of this demo, I will continue as it is, but I appreciate that you can see that this might be a potentially subpopulation, which is CD11b negative within clone 1. So the last thing that I will show you is the last visualization method, which is the XY plot. So for that, let me just show you 2 markers, which are CD34 and CD90. So this XY plot is very interesting for those who are working with flow cytometry. You are probably used to see these sort of plots. Here you can represent the expression of 2 different antibodies at the same time. When colored by sample, you can clearly see the double positive population, single positive populations, and double negative population. But now again, we have the additional analyte at our disposal to see whether there are any changes on expression values for the wild type versus the mutant cells. So that's what I will show you now by overlaying the genotype information. And this is what we get. So here what you can see, let me just focus on the wild type first. So in a background of CD90, CD34 positive cells, we see that the wild type cells, vast majority, seem to be CD90 positive. And very few of them are CD90 negative. Then some of them are CD34 positive, some of them are CD34 negative. Now, when we add the mutant population, we see that there are actually different changes in terms of expression of CD90 and CD34 within the double positive population. If I now get rid of the wild type for you to see it better, you can see that even though they are both CD90 and CD34 positive, there seem to be slight different expression values for the mutant cells versus the wild type. So the mutant cell seems to be seems to have a higher expression of CD90, lower expression of CD90 and higher expression of CD34 when comparing to the wild type cells. So I hope I convinced you that this is an easy way to analyze your multi-omics data. And, you know, I consider it's very easy to use and navigate through the different menus. You have many options for clustering, many options for representing your data, but of course you can do many more things, right? That's what Kelly was showing before. So for more advanced analysis, we have developed the Mosaic, which is Python based. And this is now what Kelly will show you. So This is the end of Tapestri Insights demo for now, and I'll hand it over to her.
Great, thank you, Hema.
Thank you.
Do you mind stop sharing and I'll share my screen?
Yeah, sure.
Okay. Ah. All right. So Hema did an amazing job taking you through Tapestri Insights software. And so my job is a little easier, explaining what Tapestri Mosaic analysis package is. So if you go to Mission Bio has our own GitHub page. So you can go to github.com/missionbio, and there you'll find our Mosaic analysis package along with other data analysis tools. And so this is based on Python code, and we use Jupyter notebooks in order to execute the code. And so you go here to install the package and upload your sample. In addition to the Jupyter notebook, we also have a GUI based on Streamlit, which allows you to take the code and look at different plots in a nice interactive fashion and turn on different DNA or protein markers of interest, all after uploading your H5 file. So with that, I'd like to give you a sneak peek of what the Mosaic Jupyter notebook can do. And so this is it can do exactly the same thing that Tapestri Insights software can do, but it has more advanced analysis tools. So for instance, one of the very first things you would want to do is identify all of the DNA variants that exist within this sample. And so we can use default Tapestri Insights filters in order to gain all of the 20+ variants that are on display when you open up Tapestri Insights software. And then we can plot these variants on a correlation plot in order to find interesting variants that may be of interest. And so here we see a correlation between the pathogenic IDH2 and the pathogenic DMT3A mutation. We know from bulk sequencing that these two mutations are important for this patient sample. And then with the ClinVar annotation that our software does, we also know that these two variants are important because they're labeled as such and identified as pathogenic versus all of these other variants that either are intronic or benign or have no known pathogenicity. So for the rest of the presentation and what we did with the analysis is we focused on these two variants and wanted to explore them further. So then you can select these two pathogenic variants and we can bucket them in a heat map based on genotype. And so when we do this, we see this wild type population at 23%. And then we see this larger pathogenic or mutated population that mostly is heterozygous for both the IDH1 and the DMT3A variants. And so if you recall going back to our UMAP that Hema showed, it corresponds to that nice purple population that was wild type and the nice green population that was mostly double mutated at these two variants. Okay. Now we'd like to explore our data and how the DNA variants correlate with protein expression. So to our heat map, we can add the protein data and keep our same buckets that we determined. So identifying our wild type population based on the wild type genotype and identifying our mutated population based on the presence of these, the heterozygous of these two variants. And so what we can nicely see in a heat map is that the wild type population correlates with upregulation of CD3, which is very strong. And the pathogenic or mutated by genotype population corresponds with upregulation of CD38, which is what we would expect. So CD38 is a stem cell marker, and has been implicated in being expressed on blast or pathogenic cells, versus CD3 is a marker of mature T cells. Okay. In addition to these heat maps where you can, I just want to also reflect, this is a lot of data. So where each row represents a cell and each column represents an analyte, we get information from the same cell across multiple variants and multiple proteins. Here we're showing a smaller data set of 2 DNA variants and 6 proteins, but it becomes extremely powerful when you can do 10 SNVs and 45 proteins. The amount of data that you get from that single sample is really immense. And you can see that reflected by each individual row. Okay. So then we have other visualization tools in our Mosaic analysis package. We can simplify the genotype of these 2 groups. We can quantify with a bar plot exactly the same as we would do with Tapestri insights. So we see that roughly 25% of the cells in this sample are wild type versus 75%, which are pathogenic. We can create Violin plots for each protein marker to get more quantitative expression levels for each protein. And so here we see with CD3, we have upregulation in the wild type population versus the pathogenic population. And if we look at CD38, we see this upregulation in the pathogenic population versus the wild type. Along with Violin plots, we have ridge plots. And I think ridge plots are really beautiful. So if we look at CD3, and this is across all cells, you can see this really nice bimodal expression where you have a population of cells that express CD3 and you have a population of cells that don't express CD3. And it's very binary, off or on. Versus if we look at these other markers like CD90, CD45RA, we see more of a continuum of expression where each cell might be low, medium, or high expression. And so I think ridge plots do a really beautiful job of depicting this, the expression level across all of your cells and whether it's binary or exists more on a continuum. And then you can also visualize these ridge plots. You can break them up by the genotypes that you labeled based on the DNA information. And so here we see the differences in CD3. We know that the high expression is in the wild type population and the low expression is in that mutated population. Okay. And then finally, so we did our demo on one sample. Tapestri Insights 3.0 can handle multiple samples, as does the Mosaic Jupyter notebook analysis package. And so here, not only did we have the one patient sample, which is shown in blue, which has these 2 DMT3A and IDH2 mutations, but actually in collaboration with Ross Levine's lab, we actually looked at 6 different patient samples, which are shown in these colors on the left. And so you can look across multiple patient samples to see what SNVs are in that sample and how that correlates with the protein expression on the right. And so if we take a closer look at the blue sample, the one that we looked at very closely, we see it's double positive for DMT3A and IDH2, and we see this nice expression pattern. If we look at the bottom, what was interesting was this was our nice positive control. So in the field, it's known that typically NPM1 mutations correlate with negative expression of CD34 protein. And so that's exactly what we see here. The patient that has an NPM1 expression has lower CD34 expression. And this actually fits really nicely with what Dr. Stephen Chung found at UT Southwestern. He gave a webinar last week. I encourage you to check it out on Genome Web. And he also shows the same phenomenon where he has a patient that has the NPM1 mutation and in general has lower CD34 expression. But what was really exciting about his data set was he actually found a subpopulation that was CD34 positive, which was unexpected, that still contained the NPM1 mutation. So I think right now we're kind of looking at this bluntly larger effects, but you can also, again, cluster within the CD34 population to find even more refined subpopulations overall. Okay. And then finally, we can simplify these heat maps, again, just based on genotype. And we can look at the expression now quantitatively across these 6 different patient samples. And so again, if we look at CD34, we see this nice downregulation in the patient sample that had the NPM1 mutation. And we see clearly now across all of the different samples how the genotypes correlate with protein expression. So again, we're combining DNA, protein, multiple samples, and integrating all of this data in easy to use visualization tools. So with that, Dr. Ross Levine and Lindy Miles, they recently published their findings using the Tapestri platform to look at DNA plus protein in a recent Nature paper. So we highly encourage you to check it out and see how they did their data analysis and see the implications of the biology that they came to based on these studies. And with that, I'd like to thank everyone for attending. We are able to today highlight the multi-omics capability of combining SNVs and proteins. We can also do copy number variation, which we didn't have time to explore today. But Mission Bio is the only platform that can integrate from thousands of single cells information about SNVs, CNVs, and proteins. So all with one simple assay, which was enabled by adding a simple cell staining protocol upstream to the Tapestri workflow. If you would like to access these software and data analysis tools, for the software, you can go to Tapestri portal at portal.missionbio.com. And there you can launch Tapestri Designer to create custom DNA panels. Tapestri Pipeline is the secondary analysis tool. Here you can download Tapestri Insights Software 3.0 in order to unlock these new tools for DNA plus protein analysis. And then finally, I showed you previously, but go to our GitHub page in order to access Mosaic. We have vignettes and tutorials in order to help you get started analyzing these very complex data sets. And with that, I'd like to say thank you, and we'd love to take any questions you have.
Thanks, Hema and Kelly. A quick reminder for the audience to submit their questions in the chat box now. We actually had a couple questions that came in. The first question is, can you quantitate the cell surface protein expression with the Tapestri Insight software?
Yes. You can. Hema showed the beautiful, so with the scatter plot, you can see differences in expression levels between your different populations. We didn't show it on this webinar, but also in the Violin plots, we showed you what it looked like looking at different DNA parameters, but you can also look at your protein expression via the Violin plots.
Great, thank you. I have another question here. Is the DNA plus protein workflow compatible with fixed cells and intracellular protein staining? So it looks like a question about some sample type.
Yeah, so fixed cells has not yet been tested, and intracellular protein staining is development work. So we have not tested these. Fixed cells has been tested and validated for DNA only, not yet for DNA plus protein. And we're working on the intracellular protein staining, but we don't know when this will be ready.
Great, thank you. How many samples can be analyzed in the Tapestri platform simultaneously?
So in the Tapestri Insights 3.0, you could add multiple time points. So the idea is that the samples you analyze together belong to the same patient. So if you have, for instance, 4 or 5 different time points, you can analyze them together, but all from the same patient. That's where you will get the majority of it, the best of it.
Got it. Okay. This is an interesting question. Do you provide bioinformatics support for these analysis tools? For people who don't have a lot of bioinformatic resource, where do they start?
Yeah, sure. I mean, we provide training for all of our customers. So we will have a session similar to the one we had today, where we take them through the software. And there are a lot of documents available on the website, as well as videos and follow-up session to help people familiarize with the software, regardless of their bioinformatics skills.
And
That's good.
I'd like to add, we have a fantastic FAS team at Mission Bio. And the Jupyter Notebook, even someone with very, medium level skills in bioinformatics, Hem and I are able to easily execute the Jupyter Notebook and unlock these advanced analysis tools. So really, we support all levels of ability.
Okay, a couple more questions just came in. This is also an interesting one. The software, does it only support CMV mutations in protein, or does it also support ATAC-seq?
You want to take that, Kelly?
Sure. I mean, at this time, we do SMVs and CMVs and protein, and we don't do ATAC-seq at this point.
Are there plans on correlating mRNA expression?
We always get this question. Yeah, so there are plans, but again, we don't know yet when this will be available. But it is the next logical question, right? As we encapsulate a whole cells, we have actually access to all the analytes within this cell. So the next logical step would be trying and tackle RNA simultaneous with DNA.
Great. Oh, here's the one. Is the Tapestri software free, or is it included in the platform?
I'll take that one. Yeah, it's free. So you don't need to pay additional anything additional to analyze your samples. It is included in the price of the reagent. So you have access to the software, all the software packages that Kelly talked about in Pipeline and Mosaic as part of your assay. You don't have to pay additional.
Yeah.
Great.
Also would encourage you, we have example data sets. So when you download Tapestri Insights 3.0, it comes with a data set, which is from our 45-plex and on PBMCs. So I encourage you to play around there, as well as with the Mosaic Jupyter notebooks. There's an example data set as well that you can explore.
Great, that's great, because that was the next question. All right, it looks like we have another question. Not sure if we can answer this. How do we normalize the protein data? I guess in the Tapestri software.
Mm-hmm. So protein expression, we use, I showed different methods. So one of them is CLR, is a center log ratio. This is typically used for site-seq. So for those using site-seq, that would be familiar to them. Or you can also use a different method like inverse hyperbolic, or which is similar to FACS, to flow cytometry. So for those familiar with flow cytometry analysis, that can be used too.
Yeah, and then I'd also like to mention that in the Jupyter Notebook, we have a third normalization tool that is specific to Mission Bio that was created by our bioinformatics. And so you can also explore that third normalization tool in the Jupyter Notebooks.
Great. All right. It looks like that's all the questions that we have today, but just want to thank the speakers for the wonderful webinar and the software demo. And thank you to the audience for joining today. And with that, I'll conclude today's webinar. If you have any additional questions after the event, please don't hesitate to contact us at Mission Bio. And thank you, everyone, and have a great rest of the day.
Thank you.